Hello everyone, and welcome to our second online seminar for National Myeloma Month. My name is Deborah, and Juliet and I, both of us myeloma support nurses, will be chairing the seminar today. To begin this event, we would like to acknowledge the traditional custodians throughout Australia and recognise their continuing connection to land, waters and culture. We pay our respects to their elders past, present and emerging. The theme for National Myeloma Month this year is Wellbeing and Possibility. Firstly, Dr. Nick Murphy will talk about new treatments. We will then have a 5-minute break, followed by Dr. Shafkat Iman discussing CAR T-cell therapy. Our final speaker today will be Elizabeth Wells, talking about cognitive impairment in cancer and the ability of reading to boost well-being, especially reading aloud. Each speaker is happy to answer questions following their presentation. Please use the live Q&A option below if you would like to ask a question. As stated, you will need to type in your name and tap join, or choose not to have your name displayed and type in anonymous. You can also use the live chat box by tapping on the 3 dots in the top right-hand corner. And there is a telephone number and email provided if you have any technical difficulties. Any questions that don't get answered today, we will endeavour to respond to within the upcoming week. The recording today, as with all our online presentations, will be available for you to watch at a later date. It usually takes a week from the live presentation for it to be uploaded onto our website. So I would now like to warmly welcome Dr. Nick Murphy, who is a clinical and laboratory haematologist at the Royal Hobart Hospital with an interest in myeloma. Nick is the Tasmanian representative of the Myeloma Australia Scientific Advisory Group and State Councillor for the Haematology Society of Australia and New Zealand. He has a young family which occupies most of his time outside of work. So now I'll hand over to you, Nick. Thank you.
Well, thank you, Deborah, and welcome everybody. I'm dialing in from a somewhat indecisive Hobart today with intermittent sunshine and gloomy clouds. My job is to talk about what's on the horizon for myeloma. I'm not going to be talking about CAR T cells, as Dr. Imam has that responsibility and will do a great job, I'm sure. So I'm not especially old, potentially I'm younger than many of the participants online, but the so-called new agents came to market around 2000, and the first of those was thalidomide, and they're now very old. I've just put in there a little bit of information about how thalidomide and its derivatives work, the so-called immunomodulatory drugs, or IMIDs for short, because we'll touch on that a bit later on when we're talking about new therapies. So traditionally, the new therapies in myeloma come down to 2 families, the so-called IMIDs and the proteasome inhibitors, or ZOMIBs as I call them, and the particular drugs are listed there. All of the IMIDs are oral, and the proteasome inhibitors are either subcutaneous, intravenous or oral therapies. So I've put this slide in, some people may not be aware, but here we have Jackson Storm, the flash new one in the dark blue and black, and Lightning McQueen, who's determined and older, but still triumphs in the end of Cars 2. I don't want to spoil the film for you. But anyway, so this year in 2022, when we talk about newer agents, we're talking increasingly about targeted or smarter drugs. I think of these like selective weed killers rather than a non-selective weed killer like chemotherapy, as well as what we call small molecules that get into the cell and disrupt the mechanisms within the cell and cause fatal injury. I'll touch on each of these families of drugs, but again, I won't be talking about CAR T cells. Here we have a nice picture that illustrates what we're talking about when we're talking about immuno or targeted therapies in myeloma. So down the bottom, we have the myeloma cell, and on its surface, it expresses a number of different proteins that can be targeted. And in a perfect world, we'd have a target that is only on myeloma cell that we can target very strongly and specifically, which means I'm not going to cause too much fallout or damage to other cells in the body. CD38 is one of the more prominent, and daratumumab was the first to come to market in Australia, but there are other agents such as isatuximab listed there. A new drug to market in Australia just in the last few weeks, so exciting new times, is elotuzumab, so another mAb, monoclonal antibody, that targets a protein called SLAMF7. Further along, we see BCMA, which stands for B cell maturation protein or antigen, and there are a number of ways in which we can target BCMA with immune therapies, as well as targeted drugs. Beg your pardon, I've just updated my phone. Okay, so how do we get these new drugs in Australia today? Outside of the PBS is what I'm talking about. So the best way, in my opinion, is through a clinical trial. And I know that not everybody is treated in a large hospital that has active trials units. So there are compassionate access programs that the pharmaceutical company make the drugs available before they come to market. I don't like to be a cynic, but they generally do this once they're pretty confident that their drug will be listed on Medicare, and therefore their $1 billion will be not too far away. But generally they try and mirror the clinical trial inclusion criteria and have similar restrictions on who can access the drug at no or significantly reduced cost. I sit on the hospital committee for my hospital. That just sort of runs their eye over these access programs to make sure that the drug won't be withdrawn from patients who might be benefiting at short notice, because that would be a major ethical dilemma. But sometimes they're open for a set period of time, and they generally don't, don't stop these access programs abruptly. If they did, or they didn't undertake to keep providing the drug until it becomes Medicare available, then often they would not get approved for use within our hospital at least. So the first newer therapy I want to talk about is daratumumab, and up in the top right you can see some brown circles, and they are CD38 staining myeloma cells within a bone marrow. So CD38 is expressed predominantly on plasma cells, which is the myeloma cell. There is some expression on lung cells and red blood cells, which can lead to some issues. But the pivotal study, there were two that ran almost at the same time, was the CASTOR study, where daratumumab was added to bortezomib and dexamethasone, or not. And we saw fairly dramatic early improvements in progression-free and overall survival. So progression-free survival is where your disease is under good control and you haven't needed a new treatment, and overall survival is pretty black and white, dead or alive. So these were quite striking results early, 83 versus 63% chance of responding overall, and some of those responses were very deep. I've got some other illustrations coming. So here we have purple line is daratumumab, orange line is not, and on the left we have overall population, and on the right we have, and this is significant because this is what got PBS or Medicare's attention, is that if it was used as the second treatment for myeloma, the difference between the purple and the orange line was more stark. One could argue that point, but it's ultimately what Medicare accepted. Around the same time, daratumumab was added to lenalidomide and dexamethasone, and the results, in my view, are fairly similar. However, the producers of daratumumab pursued listing with bortezomib only and not with lenalidomide at this stage. Outside of first relapse or second myeloma treatment, we can access daratumumab in Australia for patients who have had multiple lines of treatment. They have to have had bortezomib and lenalidomide and pomalidomide. So we're selecting out some people who've had quite a number of lines of treatment. And daratumumab is used as a single agent, although we use a very generous dexamethasone pre-medication in our institution, and I'm sure around the country do similar. So these patients have about 1 in 3 or possibly more patients will have a response. And my personal experience is that this drug provides reasonable control for people somewhere in the order of 12 months if they respond. I do have one patient who's now been on it for about 3 years, but he's my longest runner. There are some issues with daratumumab, like there are with every single drug in myeloma. The most important from the patient perspective, in my view, is the fact that it can cause inflammation in the lungs because CD38, which is the target of daratumumab, is expressed on lung lining cells. Excuse me. Most people don't have infusion reactions, but a small number do. And if we're using daratumumab through the access program, these patients are more heavily pre-treated later in their myeloma course and have more pre-existing side effects from other therapies. And so generally more kind of fragile, I suppose. Another place where daratumumab may have a role in the future in Australia, certainly elsewhere in the world, is in the first-line treatment setting for patients who are not heading to transplant. So this was a major study published a few years ago now that compared lenalidomide and dexamethasone, which is the standard first-line treatment, one of the standard first-line treatments for non-transplant eligible patients in Australia and globally, and they added in daratumumab or not. And it wouldn't surprise that adding in another drug improved outcomes. So here we have the daratumumab lenalidomide arm in the red or ochre, and the lenalidomide dexamethasone in the yellow. So patients do better, and they respond, and the responses seem to be more deep, which we know confers to a better long-term outcome. Another CD38 targeting agent is isatuximab, and we can access this in Australia, as far as I'm aware at least, only through a clinical trial. And the ALLG, or Australasian Leukemia Lymphoma Group, do have a trial for patients who commence lenalidomide and dexamethasone, and then along the way have various sort of landmarks or targets of response that they need to meet. And if they don't meet them, then we add in isatuximab to try and improve their outcome. So this trial's been open for perhaps a year or a couple of years. And there are some numbers or information coming through, and it would appear that most patients who add in isatuximab to lenalidomide and dexamethasone do deepen their response. What that means in the longer term will play out in time. So this is Graham Jackson. He's one of my personal myeloma heroes, which shows how interesting I am to my wife. But anyway, so he really, in my opinion, designed this tremendous real-world applicable trial. Some trials are less applicable to the real world than others. So patients had, all patients had treatment first line, then they got randomized to get lenalidomide ongoing or not, and then until they progressed. And it did show, and it built on some works from previous studies, an overall survival benefit by adding in lenalidomide maintenance after a stem cell transplant. And excuse me, I've done a RATCH test, not that you can catch anything through the internet. This impressed the PBS so much so that lenalidomide post stem cell transplant has now been approved for a few years, about 5 years now, for maintenance therapy post stem cell transplant. I'm not talking about CAR T cells, I'm sorry for the picture. Excuse me. What I am going to talk about is a treatment option that is available, as we say, off the shelf, and that is a bispecific T-cell engager. And I think of these like evil matchmakers. So here we have a little molecule, which is sort of teal. I'm not sure about the political implications. That on one end will grab onto a myeloma cell, and on the other end will grab onto an assassin that's going by, and will introduce the assassin to the myeloma cell and cause tumour death. I think these are extremely clever molecules, and in my opinion, potentially have a lot of advantages compared to CAR T cells. They don't need to be manufactured or engineered, which takes, I'm sure we'll hear about it later, but takes a number of weeks. And if somebody's myeloma is growing or progressing very quickly, it might be problematic. So generally faster, generally easier. Hard to believe it wouldn't be cheaper. And for those of us who don't work at the what I call mega cancer hospitals in Victoria, that's Peter Mac. Elsewhere have their own mega hospitals. Other hospitals might be able to offer this, so potentially have a more broad applicability. So the bispecific T-cell engagers were first developed for acute lymphoblastic leukemia, and that drug's called blinatumomab, and that does seem to work. They also seem to work in relapsed diffuse large B-cell lymphoma. So these are both lymphoid malignancies, as is myeloma. I think I've touched on the next point already, but there are a few target proteins on myeloma cells, and the one that's most advanced in drug development is BCMA, as I mentioned earlier, with at least 3 drugs that have gone through major clinical trials. Because myeloma is a cockroach and a survivor, many patients do relapse after a BCMA-directed therapy, so there are other agents in development targeting a different protein on the myeloma cell surface. Indecisive Tasmania has become sunny again. I'm sorry. Ignore the last line on that slide because I'm not talking about CAR T cells. So this was a slide that I acquired from the ASCO meeting mid last year, and it's just got some pros and cons for CAR T versus bispecific antibody therapies. And one could easily argue, excuse me, that while the CAR T cells have been around a little bit longer, that bispecifics have potential to help more people because there's no lead time, there's no need to engineer them. You start patients in hospital, but then after that it's an outpatient administered therapy. And there are some weird side effects of CAR T cells that I'm sure will be covered later on that tend to be less of an issue with bispecific agents. But this is not a debate. I think they're both tremendous advances. The other broad family is an antibody drug conjugate. And so again, the one that's most advanced in development is called belantamab. And on one arm has a target that grabs onto the myeloma cell, in this case BCMA as well. And then it also, that drug by binding activates the immune system to try and kill the myeloma cell, but also delivers a nuclear warhead to the myeloma cell that is lethal. And I think of this as something that, like the Snow White's apple from the evil queen, that was irresistible, and then she ate it, and then she fell into a deep sleep, but ultimately was not dead. We want this to be dead. So just for people who like graphs, belantamab was first developed a number of years ago now, or a couple of years ago now, and early phase showed reasonably good response rates. 1 in 3 patients responded to 2.5 milligrams per kilogram. And those patients were heavily pre-treated. I think that had 5+ lines of treatment. And so most patients stayed on treatment for about a year. We can access belantamab through a compassionate access program in Australia today. However, I'm not sure where this drug is at in terms of seeking PBS reimbursement. There are ongoing clinical trials involving belantamab either as monotherapy or in combination with other drugs around Australia, and it'll be interesting to see how they read out. My personal experience with this agent is that it is highly potent, but does come with some particular side effects. Particularly, it can cause irritation of the cornea and causing blurred vision. This is a picture of my oldest daughter, excuse me, sorry, who spent a great deal of time making a cake. And we've got lots of ingredients for myeloma. It's just a matter of getting it right and pulling it together at the end. So that was meant to be a seamless segue into the small molecules. The first I'm going to talk about is venetoclax, which was actually developed in Melbourne by one of my old bosses at the Royal Melbourne Hospital, Andrew Roberts, and his colleagues. It was actually developed for chronic lymphocytic leukemia. But does have activity in myeloma and paradoxically acute myeloid leukemia. Not paradoxically, surprisingly. So basically the BC ligand, BCL number 2, is a protein that's overexpressed in certain blood cancers that helps the cancer or tumour cell to survive and thrive. And so by blocking that, we make the cell more susceptible to being recognised as cancer and therefore being killed. So this graph's a number of years old now, but it did show that patients who were receiving venetoclax and bortezomib had really good responses overall. This led on to a bigger study that had some safety concerns overall, which probably, in my opinion at least, relate to infection preventative medications and their lack of administration consistently. But nonetheless, it would appear that venetoclax is most active in patients who have a particular gene change, which is called translocation 11 and 14. And these patients seem to have a more deep response, and therefore that seems to be where this drug will land in the myeloma landscape. Another small molecule is called selinexor. And this has a very novel mode of action. So it has activity in patients who have had numerous lines of treatment, some even up to 11, I read in the papers, which is pretty imaginative if you can come up with 11 different treatments for myeloma. But anyway, so patients, even heavily pre-treated and progressing on their, excuse me, I'm sorry, progressing on their previous lines of treatment, do respond. So that's promising, and that's essentially as a single agent. And then building on from that was what was called the Boston study, which included bortezomib and selinexor. And if you've got a magnifying glass, you can see the dosing mechanism. Selinexor is an oral therapy. It's given once a week, but by no means is it free of side effects, I can assure you. So there has been a selinexor access program in Australia that closed last week, but patients who were on the access program will still be granted access, and selinexor is moving towards submitting for a Medicare listing later on this year, I understand. There was a drug, and I said it was because I think it's sort of dead in the water, called plitidipsin, or Aplidin was its other name. I've only used it once, again through an access program. It was derived from some obscure deep sea algae molecule, and even when I asked the people who work for the company how it works, they didn't really convince me that anyone really knew. However, I understand there are some ongoing studies in combination with other drugs. So we're trying to scale to the great heights. That's again my oldest daughter. I should tell her that I'm using her images, but I'm her father. And this is my last sort of section, is iberdomide, which is a derivative of the imids, thalidomide and families. Basically, there's a consistent molecular structure or molecule structure in thalidomide and derivatives that seems to confer the activity, and that seems to be happening by inhibiting what's called cereblon, which is a protein system within the myeloma cell. So iberdomide basically is a more specific and more potent thalidomide family of drug with far less side effects, and the main side effects of the imids, as I'm sure many people listening will know, is peripheral neuropathy, and likewise for the proteasome inhibitors. So iberdomide shows activity again in patients who have had all previous lines of therapy, and interestingly, including people who have failed lenalidomide and pomalidomide. So there are ongoing global studies in combination with other agents. So that's another theme, is using a new thing in combination with something that's tried and true. So there are active studies using cyclophosphamide and dexamethasone, as well as what's called a backbone or platform study, where most people get iberdomide and then it's combined with bortezomib or carfilzomib or daratumumab or two of those, and then see what happens. In Australia or Australasia, we have a couple of studies that are in the wings in using iberdomide, dexamethasone and isatuximab, which makes for a nice combination logically, as well as another one that involves venetoclax, which I mentioned earlier, with iberdomide and dexamethasone. Again, that's limited to the patients who have that gene change, the translocation 11 and 14. And just, I'm trying to not say pivot, but just reflecting a little more broadly on horizons, and again, non-medical stuff here. We are improving anti-infection prevention, and patients therefore have less infections and therefore have better quality of life and actually live longer. There is this idea out there or concept about curing myeloma, and people talk about a functional cure, or functional cure is the language that gets thrown around. Sun's in my eyes, I'm sorry. And so I was at a talk a few years ago from Joseph McHale, who's from the US, and he interviewed heaps of people and patients with myeloma, and the theme that came through was, if I don't have to think about the disease or do anything in particular about it other than take my pills, then that's kind of analogous to being cured, a bit like having high blood pressure or high cholesterol. And then reflecting further, patients who have high risk myeloma don't necessarily die within 3 years. A number of patients can live for a very long time. So there's a lot of change on the horizon, and I would say this is exciting. We're in an exciting time in myeloma therapy. When I moved to Hobart about 9 years ago, nobody really wanted the myeloma portfolio because it's kind of depressing and nothing new, but things have changed dramatically, and I hope that continues. And that's really the end of my talk, and I'd be very happy to take some questions.
Thank you very much, Nick, for a very thorough and informative talk. We do have a couple of questions. The first is from Joe in WA. And look, she wants to know, how do we compare to, say, America or the rest of the world when it comes to new treatments and availability of drugs? You know, is Australia comparable, or, you know, do we lag behind?
We do lag behind. That's a statement of fact. We have a different health system, and you could look at that in different ways. I would argue that Australia, we do pretty well internationally. We are behind the US, where they have a different fee structure and funding structure. I would say that our system of Medicare is more equitable, and if a drug is listed, then more people will get it, as opposed to in America, where if a drug is listed as being able to be used, then anyone can buy it, which I personally find a little challenging. But in terms of access, acknowledging the differences in systems, we are behind other parts of the world. It's true. And that is a source of great frustration. But even without thinking about it too much, just in the last month, we've had elotuzumab in combination with lenalidomide listed. So wheels turn slow, but they do turn. And that's another reason, if you can, to enroll on a clinical trial, because that's a good way to access new drugs.
Okay, thank you. Andy is in New South Wales, and he's interested in, Dr. Murphy's, your view on natural therapies. He says there are so many side effects to traditional chemotherapy treatments. Do you think there is a place for natural therapies, you know, to help with traditional treatments?
It's one that I actually get asked quite a lot. So the two natural therapies I get asked about the most are turmeric or curcumin and medicinal cannabis. So the first one, curcumin or turmeric, I've got a number of patients who have accessed this themselves from a local pharmacy or apothecary, as they call themselves. And one seems to be doing extremely well, and the other didn't really help her at all. So there is some evidence that big whacks of curcumin slows down growing of myeloma cells in a test tube. So there might be something there. The other agent that I get asked about is medicinal cannabis, which I know is very topical because it's now available in various jurisdictions around Australia with slightly different rules depending on where you live. I would say that, to my knowledge, there is zero evidence that medicinal cannabis does anything against myeloma cells. However, there does seem to be utility for medicinal cannabis or cannabinoid derivative products for nausea. And I know a number of people find it very helpful for chronic pain, which might be useful for people with peripheral neuropathy, for example. They would be my two compounds or substances that I get asked about the most.
Here. Thank you. One last question that we have. This is from Linda, also in New South Wales. She says there seems to be many new treatments appearing for myeloma. And this is just from me. You did talk, Nick, in, you know, how people do view a cure. But Linda asked, do you really think there will be a cure one day? And how far ahead do you think that will be?
Well, that's the million-dollar question, I suppose. I don't mean to be a nihilist, but my personal view is that no, we won't see a cure of myeloma. There are small numbers of patients who have had an allogeneic stem cell transplant and survive for many, many years. There are equally other patients who have treatment and survive for many years and then die of some other cause. And you could argue, and I acknowledge the point, that they've been sort of cured because they didn't die of their myeloma. But I think if you live long enough, it will come back one day. Where we're seeing potentially a shift is with the CAR T cells, which I'm not going to talk about, but they do seem to offer very, very deep and quite durable, in some instances, responses. And potentially I could see that you have an infusion of CAR T cells and then a couple of years later you have a top-up dose or something like that, and your drug is maintained in remission. Whether you call that a cure or not, I think is semantics, and I wouldn't argue a point about what you call it. But if you've got really good control for a really long time, then it's essentially a cure. Like, you can't cure high blood pressure, but you take a pill every day and then you die of old age. So that's what we want for myeloma. It might just be an injection.
Okay, thank you again, Nick. We don't have any more questions, so we will now go to a 5-minute break, and when we will show our two new campaign videos. So thanks again and see you soon.
All the best.
Hi everyone. Last year we introduced you to 38Mate, an awareness campaign that shone a light on myeloma and the 38 Australians diagnosed each week. This year I want to tell you about our new campaign, Joyful Moments. This campaign celebrates all the wonderful things our 38 mates, their carers and our community enjoy. Maybe it's a new hobby you've taken up or an old passion that you've found a new way to enjoy. We'd love for you to share your joy with our community. Maybe you love fishing, swimming, bushwalking, playing music, or something completely different. I've been with Myeloma Australia for a number of years now, and over that time I've met so many wonderful Australians who love doing so many different things. We know that being diagnosed with myeloma can have a significant impact on your life, making it difficult to do things you used to do. But sometimes it's possible to do the things you enjoy, just in a slightly different way. So while we spend a lot of time talking with you about myeloma and all that goes with adapting your life to living with it, we want to take some time out to celebrate all the amazing things that you do when you are being you. Because that's really the goal of everything we do, to support you to spend more time being you. Joyful Moments also fits perfectly with our focus for Myeloma Awareness Month. In May, we will have seminars focused on wellbeing and possibility, reflecting the hope that new treatment advances are bringing in myeloma. We know that at the heart of wellbeing is doing more of what you enjoy and what makes you feel happy. Sharing what makes you feel good may also inspire others to take up a similar interest and spread the joy further. So over the next few weeks, we want to hear from you, your family and friends. Just complete the sentence, I'm a 38 mate and I love, or I care for a 38 mate and I love. And send a photo or video of you doing whatever it is you love to do. Simply email 38mate@myeloma.org.au. We'll then post your pictures and videos sharing joyful moments across the myeloma and wider community. We really look forward to seeing your photos and videos. Thank you.
Welcome back from our break. My name is Juliet and I am one of the myeloma support nurses based in New South Wales. Today I have the pleasure of introducing our next speaker, Dr. Shafkat Enam, to present the much anticipated topic of CAR T cells. Dr. Shafkat Enam is a haematologist and bone marrow transplant physician with an interest in cell therapies for blood cancers. He grew up in Sydney and completed his haematology training at RPA and Concord hospitals before completing fellowships in transplantation and CAR T cell therapy in London, Peter MacCallum Cancer Centre and Westmead Hospital. This expanded clinical and research experience in the application of novel therapies for lymphoma, myeloma and immunocompromised patients. He enjoys working closely with the Alfred Haematology team to provide holistic and patient-centred care. Welcome, Shafkat.
Thanks very much for the kind introduction, Juliet. I'm going to talk today about CAR T cell therapy in myeloma, which has become quite a hot topic in the field. I work at the Alfred Hospital. I've started earlier this year, working predominantly in the bone marrow transplant and CAR T cell teams, as well as in general haematology. So first I wanted to talk a little bit about the immune system, which has become well known to everyone over the last couple of years. As we all know, the immune system is a critical part of fighting infections, both bacterial and viral. More than that, in myeloma, it is actually one of the immune cells, the B cell, which becomes a plasma cell, that is actually the cell that goes awry and multiplies in the bone marrow, causing all the attendant problems in myeloma. We've known for many years that the immune system is very important in not only fighting infections, but keeping abnormal cells in check in the body, and that includes new and developing cancers. What's been tricky for many years is to figure out how to harness that immune response for means of developing new treatments, because the immune system is actually incredibly complex and hard to engineer. However, over the last 5 to 10 years, there have been several new treatments, including antibodies and also CAR T cells, which has provided new avenues to fight cancers, not just myeloma, but a whole range of cancers. So moving on to what CAR T cells actually are. So as mentioned, T cells are a normal part of the immune system and ordinarily fight infection. How they work is that they recognize foreign material, usually infected cells, through parts of their surface called receptors. These T cells not only directly kill infected cells, but help coordinate the immune response with other cells such as B cells. Chimeric antigen receptor T cells, or CAR T cells, are actually genetically modified T cells. And I'll tell you what that actually means. So what happens is we actually engineer the T cells to have a different type of receptor. And this receptor is actually derived from B cells. This receptor is introduced into the T cells, usually using viruses. And then the T cells are grown. And then these specific T cells can recognize cancer cells through particular molecules on the surface of the cancer cells. And these surface molecules are called antigens. Ergo, the name, chimeric antigen receptors. And the chimera refers to the mythical Greek creature that is a combination of multiple creatures. So these T cells are, in a sense, part T cells, part B cells. So a very clever bit of engineering. So what are CAR T cells used for now? The first disorders that these T cells have become approved for and are regularly used are actually lymphomas and leukemias. And I'll explain why. These disorders express a molecule called CD19 on their surface. And CD19 is only present on B cells. Therefore, it's been easy to actually engineer CAR T cells that will hunt down these cancer cells but not attack other parts of the normal human body. And on the top right, I have a picture of one of the first patients who was treated with CAR T cells for her leukemia. And she's 10 years out and has become somewhat of a poster child for the success of this therapy. In Australia at the moment, there are 2 approved products, Kymriah and Yescarta, and they've been available for a couple of years now at centres around the country. The expense of these therapies has meant, and the logistical difficulties that these require, means that the approval goes through a national and local panel before the patients undergo the therapy. Now, what does the process actually involve? Some parts of this will be familiar to many of the patients dialing in because cell collection is a routine part of autologous stem cell transplantation, but there are some key differences. So this type of what we call autologous CAR T cell therapy, or CAR T cells derived from a patient's own cells, first involves going on an apheresis machine, which many patients might have experienced. Instead of collecting stem cells, we collect T cells. These T cells are then sent off, and rather than just frozen down as we would for stem cells, they're actually sent off-site and manufactured into those CAR T cells that we talked about. There is a centre in Melbourne that does that, but frequently the cells are also sent overseas, usually to North America. These cells are then grown and then returned to us, after which we give 3 days of what we call lymphodepleting chemotherapy. So not chemotherapy in the order of magnitude of high-dose melphalan, but chemotherapy strong enough to allow the CAR T cells to expand. These CAR T cells are then a living drug, so they recognize the cancer cells through that antigen we talked about, multiply, and continue to kill cancer cells over days, weeks, and sometimes months. So really an amazing technology, and something that had interested researchers in all fields of cancer, and that included myeloma. So where are we up to with CAR T cells in myeloma? Going back to choosing a target. So why it's important to choose a target that is only present on the cancer cells is that these T cells will recognize the target anywhere in the body, not just on the cancer cell. So the target molecule that's become the most popular in myeloma is something called B-cell maturation antigen, or BCMA. This is present on myeloma cells, but also normal plasma cells, which is the cell of origin of myeloma, but not on other cells in the body. There are several products in clinical trials, but nothing has been approved as yet in Australia or funded. Although I'll go on to talk about one of the products which is being submitted for funding, but that process is ongoing. Nothing is available through government funding at the moment. So I'll talk about 2 of the most advanced products and why we're so excited about them. So this is a product called, and these CAR T cells have these long tongue-twisting names, idacaptogene vicleucel, or Abecma. And this is a product, again, targeting BCMA, that in its main study overseas had responses in over 70% of patients. And these are patients who'd been treated with many, many myeloma treatments. So in myeloma, we talked about lines of treatment. So, for example, Velcade, Revlimid, and dexamethasone would be a line of treatment. And these patients had had, 6 on average lines of treatment, and yet had quite significant responses to CAR T cells, including a third who'd had excellent responses, or what we call complete responses. And this is now approved in the US, but you can see that it's been approved in a select group of patients. So those who've received 4 previous lines of treatment. So you needed to have been treated for myeloma for some time usually, or your myeloma needs to have progressed through lots of these treatments on a fairly rapid basis before you can actually qualify. And this is overseas. A similar product, although the the actual CAR T cell is slightly engineered slightly differently, is cell-to-cell or CAR-VicT. And don't compare these rates of response between the two trials because the patients would have been different. But suffice to say that this CAR T cell, which also targets BCMA, had an excellent response rate, including a group of patients who had had lots of treatments and had really excellent responses. This is also approved in the US for a similar group of patients. Now, in both of these slides, I put up what we call progression-free survival curves, or basically charts showing what the long-term persistence of these responses are. Unfortunately, in myeloma, compared to leukemia and lymphoma, it doesn't seem we've reached a plateau, at least at the moment, in terms of patients having received CAR T cells and their responses lasting forever. Eventually, and that average length of time that you get out of CAR T cells is yet to be determined, but definitely in the vicinity of perhaps 12 to 24 months, depending on the product and the trial, patients do seem to have their disease come back. And this is unfortunately a common theme in myeloma therapies. We have yet to hone in on that one treatment that will get rid of myeloma for good. But these rates of response and length of responses have certainly created a lot of excitement. And so these products are moving towards regulatory approval and hopefully funding at some point in our setting. Cell-to-cell in particular is under consideration at the moment. Now, I can't just talk about these wonderful responses without talking about some of the downsides, which, it's important that everyone's aware of the actual practicalities of what will happen during CAR T cell therapy before you agree to this treatment. A couple of the main side effects include cytokine release syndrome, or CRS. And this is a syndrome that's characterized by fevers, low blood pressure, a bit like having an infection, like the flu. And that's because it happens when the CAR T cells are actually killing all the myeloma cells and releasing all these molecules into the bloodstream. That creates an inflammatory situation. It's treated with a drug called tocilizumab and steroids, which dampen down that immune response. And steroids like dexamethasone are well known to a lot of you. Neurotoxicity, or immune effector cell-associated neurotoxicity, you might hear sometimes, or ICANS, is an unusual disorder which presents with difficulty speaking, sometimes tremor, weakness, and in the severe end of the spectrum, seizures. The origin of which is unknown, but does also occur in the first couple of weeks following CAR T cell therapy. Thankfully, both these syndromes are reversible, although there is a form of neurotoxicity that presents a bit later in myeloma patients that have been treated with CAR T cells, which is still under investigation. These products are new, and so long-term effects are still yet to be completely well characterized. Other things that we watch out for, like we do with all treatments that involve some chemotherapy, are infection, particularly given our patients that have reached this stage where they qualify for CAR T cells generally have had lots of treatment. And myeloma itself, of course, leads to an imperfect immune response. Low blood counts, both from the chemotherapy initially, but also from the CAR T cells, can persist for some weeks and months after the cells go in. The myeloma plasma cells and the normal plasma cells are critical for producing not just your paraprotein that we track, but normal immunoglobulins, which are antibodies that fight infection. So with CAR T cell therapy, you can need intravenous immunoglobulin after CAR T cells if your immunoglobulins are low. And finally, revaccination, much like after an autologous stem cell transplant, is also recommended. So at the moment, there are a lot of open questions in the myeloma field about CAR T cells. It's extremely exciting, but we don't know when approval will happen in Australia and funding. These are extremely expensive and logistically difficult and time-consuming therapies. It's sort of much like it is a living drug. The process itself is different from other medications. And so the funding pathways are different in Australia, and how it's going to be set up in terms of who will be chosen to receive the therapy and who will qualify is a completely open question at the moment. Other questions include how it'll be sequenced compared to other treatments. We've seen with approvals overseas that patients need to have had many other treatments before being able to qualify for it. And where will it be best placed or best used? It's yet to be determined. There are studies ongoing, and some of you might have heard or even been involved in studies where CAR T cells are being brought closer to the front of treatments. And that there's certainly a strong rationale for that, because we know that T cell health does deteriorate over time and through many treatments. And theoretically, if we could do CAR T cell therapy early on when the T cells are healthy, that might be a better thing. Like I mentioned, long-term follow-up for these studies is ongoing. So how long these remissions will last, whether there will be a group of patients who derive much longer-term remissions is yet to be determined. And finally, what some of the longer-term effects will be. The other picture I put down here just demonstrates, the role of CAR T cells in comparison to another type of treatment, which a lot of you might have heard about or even been accessing, called bispecific agents. And these are double-headed antibodies, which basically work to activate T cells that are already in the body. So it's an easy way of bringing the immune system to bear on the cancer cell without having to go through all the complex cell engineering of CAR T cells. There's a whole range of studies involving bispecific antibodies going on at the moment, and how that will compare with CAR T cells will be interesting to see. Finally, a slide just on the long-term future, and long-term in the field of CAR T cells and myeloma is not that long-term at all, because things change so quickly. Here are some exciting things that are happening. So the process that I've described is autologous CAR T cells, or CAR T cells that are collected from patients. You can imagine that does take time, and in lymphoma and leukemia, the time gap between sending the T cells off and getting them back is anywhere between 6 and 8 weeks. That obviously does lead to delays, and more so our patients often, by the time they qualify, are quite sick and need treatment fairly quickly. So allogeneic CAR T cells, or T cells that are sourced from elsewhere, that have been engineered so they don't cause problems like graft versus host disease, are exciting. And there's a few trials, including some in Australia, that are trialing allogeneic CAR T cells. Different ways of producing CAR T cells, including rapid production, so we don't hit that delay of 6 to 8 weeks, is another strategy. And we have a trial at the AFRA at the moment using CAR T cells that apply this platform. And other targets, so not just BCMA, but other targets on myeloma cells that bispecific antibodies and other antibodies have targeted, such as GPR C5D and SLAM-F7, are ongoing. And if you think about it, if a CAR T cell can target one antigen, why not 2 or more? And so more complex cell therapy products are on the horizon. But nothing is imminent in terms of approval, but certainly some things might become available on clinical trial. So I might stop there and see if anyone had any questions.
Thank you, Shafkat. That was a wonderful presentation on CAR T. We do have a question from Val. She has asked, can T cells be made from previously retrieved stem cells?
So that's a really good question. Unfortunately, with the T cells that are being produced for CAR T cells, they do need to be collected fresh, because a stem cell product doesn't actually specifically, we haven't collected lymphocytes specifically, if that makes sense. So stem cells are different from lymphocytes or T cells. And so previous frozen down autologous stem cell products have not been used to produce T cells. Having said that, more so, some of the manufacturing platforms require fresh cells. So we can't use frozen cells, for example, for one of the two products that are available in lymphoma. There are, in the distant future, I guess, if CAR T cells become much more widely applied, you can imagine that we'll be collecting more T cells for patients ahead of time, much like we do stem cells. But at the moment, if we do need to manufacture T cells, you'll usually need a T cell collection at the time of going through CAR T cell therapy.
Okay, thank you. We have another question from Dan asking, can I receive CAR T cells after I've had an autologous stem cell transplant?
Yeah, so another great question. The answer is yes, in the sense that a lot of the patients who are qualifying for CAR T cells on clinical trials, and certainly the regulatory approval overseas, means that most people, or many people, have had an autologous stem cell transplant already prior to getting their CAR T cells at some point. Now, there are some studies that are trying to match the two together. So you have CAR T cells soon after an autologous stem cell transplant, obviously making sure that patients have recovered in between. But no, there's no contraindication to having CAR T cells if you've had an autologous stem cell transplant in the past.
Okay, thank you. Ken has messaged in to ask, how long do you think this treatment will last before relapse?
Yeah, so it's a good question. And I guess in a sense, the million-dollar question, we just don't know. On the basis of the studies that are being, the early data that's being released at the moment, and I don't want to be deliberately vague, but I just have to be, it looks like remissions are lasting in the vicinity of months, and sometimes that those months can be 12 months or more. It's certainly not years as yet, although for the occasional patient it might be. We haven't followed up patients long enough to know. And there'll be the occasional patients who, as with all other myeloma therapies, the disease is just so aggressive that CAR T cells either don't work at all or work for very transiently, so weeks to months. So I can't give an exact number on it, but we are hoping that it's something like 1 to 2 years on average, but it will really depend on the final outcomes of the studies that are ongoing.
Okay, thanks. Joanne has messaged to ask, how long do the neurological side effects tend to last?
Yeah, so that's a great question. So there's the traditional neurotoxicity, or ICANS, that's seen in lymphoma and leukemia, and also in myeloma patients. It tends to be transient, so it goes away usually within by the 1 month stage, and often within those first sort of 2 to 3 weeks. So it comes on. So a typical presentation might be a slight headache, a tremor in the right hand, difficulty with handwriting, and some confusion that resolves over a couple of weeks. Certainly by 1 month or 1 to 2 months, we would hope that that syndrome has resolved. Now, there has been the occasional case reported where these effects do persist, and there are some late presentations of neurotoxicity that seem to present a bit more like a Parkinson's-like disorder with rigidity that have been described, particularly in the myeloma space, that are still being that are still being worked out as to why it's happening and how reversible it is. So I would say that in almost all cases, it does resolve fairly quickly within weeks, but we just don't know whether in an occasional case, some of these effects might be long-lasting. And again, in these trials, the numbers are fairly small compared to myeloma studies involving medication. So until we've collected more data, I don't think we'll be able to definitively say whether in some patients there might be longer-term, more subtle, long-lasting effects.
Okay, Thank you. And just a last question from Don. He has just asked if you could explain what is the rationale for the current MSAC submission for CAR T cells to only be available after 3 lines of previous treatment?
Yeah, good question. I obviously don't make the decisions both from a funding point of view and from the drug companies, but certainly speaking in generalities, it would certainly be a question of availability and cost. So being able to deliver a complex therapy like this requires a lot of capacity in hospitals, and more so if it was offered to everyone in the frontline setting, as in, straight up front when the myeloma is diagnosed. One, that doesn't reflect the clinical trials that have been done. And 2, it just wouldn't be feasible from a logistical point of view for anyone in any country. And so what I can say is I think the MSAC submission mirrors what would be realistically approved, at least initially. Now, in the future, if it seems like CAR T cells should actually be used a lot more like transplant is used at the moment, and capacity is slowly built across the country, then certainly that criteria might change. But I can see why that up front they're the criteria that are being proposed, and it mirrors what we actually limited by in lymphoma and leukemia. So patients needed to have failed at least 2 previous lines of treatment in those disorders before they qualify for CAR T cells.
Okay, thanks so much for explaining that to everybody. Do you think everybody will receive CAR T cells at some stage?
Look, as someone with an interest in the field and having seen some of the responses, I would certainly hope that it becomes democratic enough such that we can provide it to a lot more people than we do at the moment. I don't know whether everyone will receive it, but I can foresee in the future that a lot more people will receive it than are at the moment, because it's certainly an exciting technology with really high rates of response in people who otherwise have very poor risk disease. So that's my take on the future.
Great. Thanks so much for sharing your ideas and your knowledge with us today. I'm going to hand over to Deb now, who's going to introduce our next speaker. Thanks, Deb.
Thank you very much. So hi again. Please welcome our 3rd speaker today, Elizabeth Wells. Elizabeth is an enthusiastic researcher, librarian, and former teacher, passionate about developing a love of reading and learning in others, improving literacy, and promoting the value of reading and libraries to the community. During her decade working in public libraries in central Victoria, she noticed a frequent correlation between undergoing cancer treatment and losing the focus necessary to read for pleasure. Elizabeth has undertaken a PhD to explore how reading aloud to cancer patients undergoing treatment might influence emotional well-being. Prior to moving to Adelaide to undertake this research, at a time when libraries suffered COVID-related closures, she worked in palliative and aged care. As a cancer survivor herself, she is able to empathise with her research participants. Elizabeth has been speaking at cancer support groups around Adelaide, and in recent weeks has appeared on television and radio to discuss her research. So over to you, Elizabeth.
Thank you, Deborah, and thank you everyone for inviting me today. My name is, Deborah said, Elizabeth Wells. I am, as you know, a PhD candidate at the University of South Australia. Cancer-related cognitive impairment, or CRCI, is an important part of my research. I'm finding this research really exciting, and I have a team of great supervisors here, and I am grateful for the opportunity today to talk to you, so thank you. So cancer-related cognitive impairment, or CRCI for short, it used to be known as chemo fog or chemo brain. But now that we know more about it and who gets affected, we generally call it cancer fog or brain fog now, which we'll talk about as we go along. So doctors and scientists have made great strides in developing new treatments for cancer. but cancer-related cognitive impairment still remains a bit of a mystery. So who can get CRCI or cancer fog? Some of you watching and listening today may be experiencing it. CRCI is commonly reported occurrences affect more than half of cancer patients during and often after chemotherapy, targeted therapy, hormone therapy, immunotherapy, all the therapies that are in the cancer domain. The prevalence of CRCI varies according to different studies, each looking at different types of cancer and different types of treatment, with the most frequent occurrence seen at 75% for breast cancer patients who received chemotherapy. The studies I'll reference today are from a variety of cancer types. CRCI can present in a number of ways, but it's commonly experiencing difficulty with concentration and processing. So in real life situations, that can mean trouble reading, driving or navigating, difficulty finding the right word during a conversation, or just keeping up with conversations. People can also struggle with multitasking, feeling confused, forgetting names or dates or things that you'd normally remember. You might struggle to learn something new or just feel terribly disorganized, as though you're just struggling with normal day-to-day things. So the most impaired cognitive functions are attention, memory, processing speed, and executive functions, which are organizing, multitasking, planning, and motivation. Okay, if only it were this simple, there is no effective test. We haven't found a test yet to screen cognitive abilities with high enough sensitivity and reliability. So looking at the two pictures on the screen, the picture on the left is someone doing a test in a quiet environment. They're sitting by themselves, there's no distractions around. So in that situation, they can perform pretty well. But real life is more like that picture on the right. There are other people, there are noises, there are distractions, there's busyness. And those are generally the situations where this cognitive impairment is felt at its worst. Not necessarily a shopping centre, but just real life where there are other demands, where we're not just doing one thing quietly at a time. So health professionals have tried using standard cognitive tests, but they're just not subtle enough to make a diagnosis. So exact causes of CRCI are not known, but recent studies implicating the body's own tumour response now. So tumour development can possibly, sorry, I'm starting again, I'm not a public speaker. So the body's response to tumour development as a cause, and that's only worsened by receiving treatment. So there was a study recently where a group of mice were given breast cancer and their cognition was tested. And they are found to be impaired from the time when they were healthy to the time they had the breast cancer. And then when they gave these same mice chemotherapy, it was found their cognition was affected even further. So 3 separate brain changes have been identified as possible causes. So it's inflammation in the brain's nervous system resulting from the body's own attempt to stop the tumour growing. There's the production of new brain cells in the regions associated with memory that are limited by chemotherapy drugs. There's the combined effects of chemo and tumour development that diminishes the learning and memory regions of the brain. We know this, but there are only some possible causes, and all treatment types and even cancer without treatment, as we've said, have been looked at for different ways this impairment might occur. So looking at this next slide, I know there's lots on the slide. It looks a bit blurry actually, but hopefully you can see enough. So these are all possible mechanisms for CRCI. So looking at myeloma, the typical treatments are, chemotherapy, stem cell treatments, radiotherapy, and now immunotherapies. And you can see there are a few theories about why this is happening with those treatments. So aside from those ideas as well, there's also patient-related outcomes as well. So general health status, level of distress. There is a link between distress and cognitive impairment as well. Some studies are looking at genetics as well. So there are people getting CRCI, and lots of them, during treatment, but there's also a subgroup of patients who experience a delayed onset of cognitive impairment too. So this figure's from a study that was done last year. So if anyone would like it, just get in touch with me later. The good news though is that, from the other slide, the impairment is usually temporary. But how does CRCI impact people? First and foremost, it's frustrating. Imagine all those impacts: trouble navigating, driving, forgetting things. Frustration is a natural result, and ongoing frustration can affect your self-confidence, your quality of life, potentially your ability to self-care. You may be forgetting things. In the workplace is another kettle of fish. I spoke to a lady recently and she thought she would get sacked because she knew she wasn't performing at her normal level while she was going through her treatment. But of course she wasn't, they're not allowed to. And she did tell her boss why she was struggling, and allowances were made, but she was feeling inadequate. So that can affect workplace relationships and just her level of satisfaction with herself. Likewise, other relationships that we have in our lives can be affected as well. And this is exhausting as well, this level of confusion, the fatigue that is a very common accompaniment to CRCI. And all this can take an emotional toll. So as I said, it is usually temporary. It does get better with time. And most people say they notice improvements within the first year after finishing treatment. Other people do experience longer term effects. So what's actually happening in the brain when this is going on? Anecdotally, it seems that in years gone by, doctors didn't believe or they downplayed patients' claims of cognitive impairment. Even when patients reported they couldn't read, they couldn't remember words, they couldn't make decisions. More recently, though, research has been able to demonstrate some physical changes that can occur to the brain as the result of treatments, with chemotherapy being the worst. And the changes are principally to the frontal areas of your brain. And in the picture, that's the big tan section on the left, but it's in the front of our heads. And this frontal area is the most common region for injury in traumatic brain injury. And so from that, we can see that how important it is, that part of our brain, for expressive language and for managing high-level executive functions like organising, planning, and motivation. Also, memory and attention. I'm going to mention the temporal lobe now while we've got this picture up. And that's the bit darker brown section in the picture. And it's running lengthwise. There's one on each side of your head. They sit behind each ear. So they're used for processing auditory information or things that you hear, and also for the encoding of memory. Ways to cope. Use basic organisational tools like a diary, sticky notes, reminders on your phone. Always put your keys in the same place to avoid the frustration of forgetting where you left them. And don't be embarrassed to use ideas like this. They're strategies that will help during this time. Studies have shown that physical exercise has many, many benefits during treatment time and afterwards. The benefits are not just physical health, but also mental health. And some benefits are specific to helping with CRCI. But with myeloma, of course, check with your treating team first before undertaking any new exercises. That's really important. There's actually a really good fact sheet about exercise with myeloma on the Myeloma Australia website too. And mental stimulation. If you can read, it's great to do. If you can't, keep listening here, and we're going to talk more specifically about that in a minute. Do crosswords or word puzzles if you enjoy them. Keep learning a new language if you were learning one, or playing your musical instrument. Do Sudokus if you like them. But don't torture yourself by saying things like, oh, I must do a crossword each day to help my CRCI. Because if you don't really like crosswords, that's no good. It's important to do something that you enjoy. Otherwise, you're just going to get more frustrated. Meditation in cancer patients has been shown to improve verbal memory, short-term memory, and processing speed. So that's something to consider. Now, there are a couple of possible drug options for CRCI, but there's been mixed results, and that is something to chat about to the doctor. Likewise, cognitive rehabilitation and training may be an option, but again, that's a chat with your doctor about those ones. You might come to recognise the things that you have trouble with more than others. For example, you might find you're especially having trouble with navigating. So don't put yourself in tricky navigational situations. If you have to go somewhere potentially confusing, get someone else to navigate. And sometimes you just need some normality. Tell your friends what you're going through with your CRCI. One lady told me that her cancer fog made it impossible to go out with her friends in a group and contribute to their conversation because she just couldn't keep up with the conversation. So going out one-on-one was easier for her, or going out with the group, but choosing to sit back and listen and just enjoy being with them. And she had told them ahead of time and they understood. So please experiment with the things that work for you. There are studies going on at the moment about CRCI. Scientists are on the ball with this now. And there has been a number of theories about why some treatments are penetrating the blood-brain barrier. For instance, the cell size of chemotherapy theoretically shouldn't get through that blood-brain barrier. So now some studies are looking, for instance, at gut health, which we know takes a beating during chemotherapy, and it's thought perhaps that because the gut is so upset, it's sending out inflammatory markers into our bodies, and it is affecting the blood-brain barrier. It's making it less effective, and thus chemotherapy drugs are getting through where they shouldn't. Potentially, this is all theory at the moment, and then scientists are investigating. And when looking at all the treatment types as well, those scientists are considering genetic biomarkers and their effect on neuroplasticity and ability to repair. I went looking for some resources to recommend to you. I'm a big fan of cancer councils in Australia. I think they do a great job with everything from having experienced nurses available, but of course you have your own wonderful myeloma nurses. But they also are great for your loved ones to call and chat to. They fund ongoing research and provide lots of resources. There's a link for the Cancer Council on the screen at the moment. It's a podcast. So Julie McCrozen, she's a cancer survivor as well. She does a fantastic podcast on cancer fog. And if you scroll down past the transcription of her podcast, there are amazing links to other fact sheets and webinars. And also, if you have a look at the Myeloma UK website, that has some really good information and further links. But tell your doctors and nurses that you're experiencing this. So talk about it at a support group if you have one. Get tips and share tips with others. Remember, you're actually going through a lot at the moment and you're doing great. So remember, this will pass. Nobody can predict how long it will last. So please make some time to do the things that you love. Stop and smell the roses, pick some daisies, but please be kind to yourself at this time. But why am I here today talking about cancer-related cognitive impairment? Why am I so interested? So I worked in public libraries in central Victoria for a number of years. And during this time, I noticed a pattern emerging of some of my regular readers who lost the ability to focus sufficiently to read for pleasure once they were diagnosed with cancer and undergoing treatment. So now I'm here in South Australia looking at why that might be the case and what we can do about it. We know there are so many health benefits to reading, but we also know reading can be impacted by cancer-related cognitive impairment. So I'm interested in finding a way to restore these known benefits of reading to people undergoing cancer treatment. And given we know about the areas of the brain most impacted by CRCI, I'm exploring the idea of listening to stories rather than reading to yourself. There's no research currently on this. So the term given to this idea of reading for therapy is bibliotherapy, with biblion meaning book, therapia meaning healing in Greek. Bibliotherapy is simply about reading for its health-giving benefits. The idea is as old as ancient Greece and ancient Egypt. And the term was first coined in a newspaper article about 100 years ago. It was about a fictitious GP. It was written up in a story to try and explain this whole reading therapy idea. And bibliotherapy really developed and matured with collaborations between doctors and librarians during World War I. And it came to prominence for a time as a valid therapy, both at the front lines and in hospitals. Libraries sent millions of books to the front lines, and they tailored selections where they could for fiction, non-fiction, and magazines. And the importance of reading to morale was recognised to such an extent that some regiments even attempted to locate little printing presses and make their own little magazines or newsletters with comics and stories. I recently read a novel called The Paris Library based on a true story. And these librarians alone delivered over 2 million books to soldiers during both World Wars. And they hand-delivered books to Jews in Nazi-occupied Paris in World War II. Such was the benefit that was associated with reading. And of particular relevance to us is thinking about the soldiers in the hospitals. So they might have had 150 soldiers in one ward, and they were all in pain, they had no privacy, their spirits were low. And the librarians made sure they had books because reading offered an escape. It gave them something else to think about. They called it a privacy of the mind. So in these intervening years to the present day, bibliotherapy has been used in a variety of ways in hospitals, in libraries, and in schools, and it's been used to help both adults and children. It's widely known in the UK. It's even national policy in Wales, but it is little known in Australia. So bibliotherapy has been studied during the last 2 decades. Studies indicate benefits to all these mental health conditions, as well as to chronic pain, which is of particular interest to us, and to sleep problems. Okay, so this idea of telling stories and listening rather than reading to yourself. People have been telling each other stories for eons. This need to communicate stories is embedded in us. It's part of our very fabric of human connection. As we know, oral storytelling is part of cultural traditions around the world. And prior to the late 17th century, reading was actually a social activity. Books were read aloud rather than silently. Adult story times exist or have been trialled in a handful of libraries around the globe, including at Melbourne's library at the Dock. And I was lucky enough to hear a librarian speak at a conference about this. And she spoke of a lady who used to come down to Melbourne for her cancer treatment at the Peter Mac Cancer Centre. She was a regular attendee at these sessions, and she was also a lady who had lost her ability to focus on her own reading because of her treatment. But she came to every session when she was in Melbourne because she loved it. She felt that she could sit and relax and listen to the story and take it in and enjoy it. And hearing this was an important piece of this whole puzzle as the idea for my research grew. So thinking about different parts of the brain and the fact that the temporal lobe, these side bits, aren't as affected in CRCI as the frontal lobe. And we know some people remember a story told to them for much longer than the story that they read to themselves. This has been shown, for instance, in people with dyslexia. So reading aloud offers a human connection. It's more accessible than reading to yourself when you have cancer-related cognitive impairment. So integrative therapy or holistic cancer care includes lots of evidence-based complementary therapies. It's unclear from literature how often bibliotherapy is made available as one of these treatments for cancer patients. It's not found on any of the websites for Australian cancer centres. And from my experience, I've never seen it as an option. But like art therapy and music therapy, it would be really good to see it included. So we connect to stories and to storytelling. We know that reading is beneficial, whether it's for pleasure or for learning. And we know reading can be impacted by the cognitive side effects of cancer and its treatments. So my research is attempting to reconnect people and stories and reclaim the benefits of reading in the process. I still need more participants too for my research. So if you live in Adelaide, we'd love to come and read to you, or you live in the country, or if you live interstate, and I don't have a reading volunteer near you, we'd still love to read to you. We can use Zoom to read. So please get in touch with me if you'd like to participate or if you'd just like to find out more. Thank you very much for listening. Any questions? My contact details are on the screen, so please get in touch if you'd like to know more.
Thank you so much, Elizabeth. It is a really, really interesting topic. And thanks for sharing and informing us about your research.
Thank you.
So we do have a few questions. So Daniel asks, wouldn't lack of good regular sleep due to steroids and general stress also be a significant contributor to CICI?
Absolutely. I mentioned distress, but yes, stress. And as we know, lack of sleep, that was a torture during the war, sleep deprivation. So I agree 100%, Dan. It's not going to help you at all, lack of sleep. Maybe, yeah, give us a call. We'll come read to you instead. You can doze off while we read.
Okay, look, thank you. I just wondered myself, this is such an interesting topic. Elizabeth, and you say that you're a cancer survivor. Did you notice this yourself? I mean, how did you go through this yourself when you had cancer, or did other people tell you, or how did that?
No, that's a great question though. So the first person I saw this in was actually my mum. She had a very rare cancer, and she was hit with a lot of different types of treatments because it was so rare. It wasn't really known what to do. And she was the first person I ever encountered who lost this ability to read. It was some years later, I went through my own cancer journey, and I must admit, I was very lucky to not have chemo, because as we said, chemo is the most common way to get this. So I actually didn't notice it in myself, unless it's just my general disorganization, because I have sticky notes and diaries, and I do write things down. That could be just that, or my personality type, I'm not sure. So I was still reading through my treatment, but I've seen so many people that can't, that I formed this pattern. But as I said, I didn't have chemotherapy or targeted therapy or immunotherapy. Yeah.
Okay, thank you. No, it's really interesting. Tina asks, are there specific foods that could help?
Couldn't tell you, Tina. I do have a colleague at uni who was looking at diet and CRCI, and his research found it wasn't so much the foods, it was the CRCI impacting the ability to comprehend recipes for healthy cooking and healthy eating. So again, it comes back to the reading thing and the focus and the concentration. So I found that people were maybe not eating as well as they normally would going through CRCI, rather than specific foods.
Okay, yeah, no, interesting. Look, this is a couple of questions in one. So are there particular websites that health professionals can go to do cognitive training or cognitive exercises?
It would be, but that's beyond the scope of my research. I know there are cognitive rehabilitation programs. I'm not sure if they're in Australia, actually. That would be psychology. Yeah, psychologists would know that one. Yeah, sorry. Yeah.
No, that's fine. Thank you. And just one last one that I have. So if you're exploring something like art therapy as a form of mindfulness, can that also help to improve cognition?
Nobody's measured it improving cognition. They've measured it in terms of overall well-being, in terms of making you feel better, which is very important. It's not to be underestimated. So art therapy studies, just like music therapy studies, poetry therapy, our study, there are some standard well-being questionnaires that you implement at the start of a program and at the end of a program, and you measure the difference. And studies around art therapy and music therapy have found, yes, people are feeling better through doing these programs. So it's the hope that bibliotherapy will have a similar effect.
Lovely. That's lovely. Look, thanks again, Elizabeth. It really has been terrific to have you here and joining us in the seminar today. So I will now hand over. No, thank you. I will now hand over to Juliet, who will close today's event. Thank you.
Thank you, Deb. A very big thank you to our speakers today, Dr. Nick Murphy, Dr. Shafkat Enam, and Elizabeth Wells for donating their time for our online seminar. I would like to thank you all for joining us today. We hope you've gained some valuable information from our speakers. If you could please take the time to fill out our evaluation form below. This feedback allows us to know what information our myeloma community is wanting to know and allows us to improve on how we provide this to you. If you haven't already, take the time to have a browse around our website, www.myeloma.org.au. We have a range of resources to help you, your friends and family navigate your way while living with myeloma. We have a fantastic new podcast called My Conversations with many guest speakers, and you can find this on our website and all podcast platforms. Lastly, I would like to remind you to consider a submission to MSAC for the current proposal for CAR T cells in Australia, and be sure to send in your photos of your joyful moments. We would love to see them. Thank you for joining us.